This is Bio 181, Chapter 5 on the membrane. Key concept that we're starting with is on the cell membrane, the fluid mosaic model. So this is the model that describes the cell membrane, and it's semi-permeable, which means that only some things can go through, straight through. Some things will require a protein to go through, and some things are just not going to go through. So it's semi-permeable. Permeable is to allow things through. It's made out of phospholipids. So this is the lipid that we learned in the last chapter. And I'm going to come down here to the model and just draw one. So here's one phospholipid on one side, and then here's another one opposite it. So this is a phospholipid bilayer. It has one phospholipid on the outside and one phospholipid on the inside. So there's two total. So it's a bilayer, 2 molecules thick, and it has large floating or anchored proteins. So the proteins, let me just go to yellow here. So here's a protein. This is actually a protein. It's really large, right? And it's in the membrane. So it's crossing the entire membrane. And it's actually going to act as a channel, allowing things to go through this internal open space. Other proteins, like over here on the right, this one is basically acting as an anchor for this molecule here on the edge. That's a signal molecule, and it allows other cells to recognize this molecule. Other proteins, it allows other cells to recognize the cell. This molecule here, which you can kind of see is a protein embedded in there, also has a portion that's sticking out. And this can also serve like to stick onto other cells, right? So proteins, much bigger, they're like icebergs in the sea. The phospholipid itself, so this in the previous slide, we had one on the outside of the membrane and one on the inside of the membrane, okay? This is the phospholipid over here on the top. This part of the head is polar, which means this part is going to be attracted to water. It is hydrophilic. So it likes water, okay? So it likes water, okay? Whereas this part down here is hydrophobic. It does not like water. It's scared or phobic of water, right? Water fearing. So no water. And if you look at the molecule itself, so down here, all along here, these are all carbon and hydrogen bonds, which are all non-polar. So this region of the molecule is non-polar. So again and again, you're going to see this pattern of non-polar associated with being hydrophobic and repelling water. Okay? Whereas if it's polar, it's going to be hydrophilic and it likes water. So what's shown here is actually, if you put a whole bunch of these molecules, here's one I'm drawing up here in the upper left, then here's one on the inside. If you just dump a whole bunch of these molecules into water, they will actually form into a sphere if that water moves. So they are going to self-organize based on the chemistry into this layer, this phospholipid bilayer. And so this is how the first cell membrane really came into being, is that it had this property where part of the molecule liked water, part of the molecule didn't, and then there were enough of those that they organized and they actually formed into a sphere, and that separated the inside of the cell from the outside. Okay? This membrane is asymmetric, right? If something was symmetric, it would be the same on the outside and the inside, symmetry. A in front means not, like asymptomatic means you're not symptomatic. Okay, so asymmetric. That means the outside, so this part up here, looks different than the inside. And so the inside is often going to be anchored to, actually, these are cytoskeletal elements. So they're part of the cell structure. They're often actin filaments that are going to be connected there. Whereas the outside is going to have more of the signaling type of molecules that are going to let other cells know what this cell is. Okay, membrane proteins. So these proteins that are in the membrane, they have many functions. They can act as enzymes. Now, enzymes speed up the rate of reaction. So they speed up reactions. They act as a catalyst, right? They're a catalyst. The proteins can also be attached to molecules that allow them to be identity markers. So white blood cells are going to be going all over your body looking for foreign invaders. And you have to have this ID card on the cell, or these white blood cells will maybe attack it and kill it. Signal receptors. So these are molecules that are going to actually receive the signal. Let's see, I'm looking down here now. So over here, this is the signal receptor. So here's the big protein, and then here's the signal, these little triangles. So the triangle bonds to, and it locks in place with this protein, and then it might instigate an internal response, okay? The enzyme over here, that's shown in this one, okay? I'm just going back because I missed illustrating that. So the enzyme here, that is this one, okay, where it's actually catalyzing this reaction that's breaking down this initial reactant into small pieces in this case. And then the identity marker, okay, that would be over here, this one with this special sequence sticking outside. This is like its ID card, okay, that it's going to show to other cells and that might be the same type of cell that would bond to it, or to white blood cells. That's this. And then there's transporters. The transporter, let's see what color I could use here that I haven't used. Well, it's purple, so let me try that. So the transporter here, this channel, it's going to transport molecules from the outside to the inside. Okay, so that's a transport molecule or protein. Here, that's this one here. In this case, the example is ion. And you should remember, hopefully, what an ion is. It's a charged atom. And so it could be open all the time, allowing things in and out. Or in this case over here, this is the same type of channel, but it opens and closes. So this is called a gated channel. So these are both for transport. And then the last one is an anchor. So the anchor, again, over here on the right side in red. So this could be attaching it to another cell. Okay. All right. This is just an example if we zoomed in of the actual protein. So this is a smaller one. So the phospholipid bilayer, here's one phospholipid, here's another phospholipid. There's a whole bunch of these, right? Bilayer means we've got two molecules thick. The protein here, just wanted to show you this if you happen to see it in the book. This molecule here is the protein, okay? And this one's a little bit smaller, but you can see it's got this shape here, this alpha helix. Now that was part of the protein structure, the secondary structure, right? As long as the beta pleated sheets shown here, right? This is, again, beta pleated sheet, just as a review from the protein structure. Okay. All right. And this one is just anchored in here. Sometimes these can, it's anchored in place, I should say, by the fact that this blue part of the protein happens to be hydrophobic. So the amino acids that are in that sequence, okay, they're amino acids that don't like water, right? So they are repelled or repel water. Okay? So they just want to hang out away from water in the middle. Whereas the regions on the edge here, these are going to be more regions where these amino acids are maybe charged or polar. Okay. So they're going to want to be with water. So just this natural chemistry is going to help organize these molecules. Okay, so we did proteins, we did phospholipids. The last part that I'm going to cover for the membrane are carbohydrates. So carbohydrates, third major component, and they're really going to be critical out here to this part where they're going to be involved in like an identity, right? The ID. They can also be involved in attachment. So they're kind of sticky. So like if you think of sugar, right? And so it can also be involved in attachment. All right.